Hey, I'm Alessandro, one of the 13 passionate early-stage researchers in the European Funder Consortium SimpleSci. Together, we are trying to tackle synaptic dysfunctions in neuropsychiatric disorders from different perspectives. I'm conducting my research at the École Polytechnique Fédérale de Lausanne, under the supervision of Professor Carmen Sandi and Dr. Simona Stori, working on a new model of depressive-related disorders.
But why is it important to study depression? Well, depression is one of the main causes of mental disability worldwide, and it affects almost 300 million people worldwide, 40 million of which in the European Union only. The causes are complex and come from both social, psychological and biological factors. Some treatments are available. However, not all of them are effective in every patient, and in the EU is estimated that 3 out of 4 people suffering from depression are not receiving adequate treatment. In addition, most existing treatments have severe side effects. This is why it is so important to study neural mechanisms that cause depression in order to develop new treatments that are more efficient and have less side effects. Reduced motivation and lack of pleasures are typical features in depressive-like disorders. Multiple brain regions are involved in the regulation of motivation, but there's one particular region that draws scientists' attention: the nucleus accumbens. The nucleus accumbens is deeply located into the brain and gets engaged to produce actions directed to obtain rewards. Indeed, the nucleus accumbens has an important role in motivation, effort, reward, and vigor of a response. If it does not function correctly, it will lead to, among others, loss of pleasure, anergia, and low motivation. This is associated with several mental disorders, such as depression. Just like the rest of the brain, the nucleus accumbens is composed of neurons, the brain cells, and the neurons are composed of many elements in order to function properly. One of these elements is the mitochondrion. Mitochondria are tiny factories that produce the energy essential for the cell to function. Colleagues from my lab have recently found that one specific component present on the surface of the mitochondria in the nucleus accumbens neurons, a protein called mitophysin-2, has an important role in motivation and coping with stress. This protein is also very important for the functioning of these neurons and plays a major role in the mitochondrial life cycle. Specifically, subjects with naturally low levels of mitophysin-2 in the nucleus accumbens were more anxious than subjects with normal levels. My goal is to understand the role that mitochondria and mitophysin-2 play on brain activity and animal behavior, to develop new ways for the treatment of psychological conditions. For this, I will characterize the impact of loss of mitophysin-2 in the nucleus accumbens. To do that, I will employ a mouse model which was genetically modified to lack mitophysin-2, specifically in the nucleus accumbens, and compare it with animals with normal mitophysin-2 expression. I will then investigate how the lack of this protein affects the functioning of nucleus accumbens neurons in 3 main steps. First, via a technique called electrophysiology, with which we can measure neurons' electrical properties, like how much the neurons are active. I will investigate the effect on neurons' functioning and communication. Through microscopy, I will study the impact of deficiency in mitophysin-2 on the structure and complexity of the neurons. Finally, I will perform behavioral experiments to investigate if the lack of mitophysin-2 affects the animal's motivation when coping with stress and during social interactions. With these experiments, I hope to be able to understand the role of mitophysin-2 in motivated behavior and depressive-like conditions, hoping to shed new light on the mechanisms of depression and to pave the way for new treatments.
Thanks for watching, I hope you enjoyed the video. If you would like to know more about the consortium and the other projects in the consortium, please check our website and the official social media channels.